Now listen, here's where things get spicy, okay? Over here, we've got a growing DNA strand or a growing oligonucleotide strand, right? So for instance, this C is for cytidine. Remember, since it's a DNA strand, it's going to be the 2' end. There's no oxygen. It's deoxycytidine monophosphate, 1-phosphate, right? What's happening here? It's connected, this nucleotide is connected to this nucleotide via the phosphate of this nucleotide, right? And this nucleotide is connected to this nucleotide, again, through the phosphate of this nucleotide. Now, notice that over here, the hydroxyl group is free, right? At the 3' end, because this is 3' here, and 5' here, the phosphate is also going to be free. Now, suppose that this is a growing DNA strand, you know, synthesis is going on, and now what we want to do is we want to insert this thymidine triphosphate. Notice how it has 1, 2, 3 phosphate groups. Again, the first is going to be called alpha, the second is the beta, and the third is the gamma, right? So this is, and since it's of course going to be thymidine triphosphate, we don't have to say deoxy, because again, remember, thymine is only going to be present in DNA. Anyway, so we want to take this structure right here, the thymidine triphosphate, and we want to insert it into the growing DNA strand. So how does that happen exactly? How do we do it? Now, look here. This is going to be, so we took this, we magnified it, we put it right here, right? See, the deoxyadenine monophosphate, and now I want to link these two with one another. Now, if you notice the phosphate here we just write it PPP for simplicity, okay? Actually, what's going on here is that the chemical structure of phosphate is going to have a hydroxyl group right here, right? And it's linked to the carbon of the nucleotide via an oxygen, okay? So how do we take this and take this and we just add it to the growing nucleotide strand? So here's what's going to happen. The hydroxyl group of the phosphate is going to interact with the hydroxyl group of the 3' end, or in better terms, the 3' end here of the hydroxyl group on the 3' end is going to attack the alpha phosphate, which is this phosphate, the first phosphate, right? Now it attacks it where? It attacks it at the hydroxyl group as well. So what do we get here? What do we see? Basically, if we take both of them right here, you've got a hydroxyl group which comes from the 3' end, and it's, attacking the hydroxyl group that's coming from the phosphate, right? And so basically, what's going to happen is that this hydroxyl group is going to be thrown out. Along with it is going to be this hydrogen group, right? And so, you've got the hydroxyl group and you throw out another hydrogen with it, and now what do you have? You have water. This is H2O, okay? So we have H2O, which is water, and this is exactly why we call this type of reaction that occurred here a dehydration reaction, because we dehydrated the hydroxyl group from here, and we produced water. They may call it condensation reaction, by the way, also, right? Anyway, what's the end result? After we took this, and we have our water right now, right? And we took the hydroxyl group off this phosphate, and we removed the hydrogen. And so the 3' end of this nucleotide is only going to have an oxygen, and of course, at the same time, to drive this kind of reaction, the condensation reaction, the dehydration reaction, these 2 phosphate groups are going to be released, and we call them the PPI, or inorganic phosphate. When they're released, they're going to give a massive amount of energy, which drives the condensation reaction. The end result is that the oxygen here that's present on the 3' end is going to link to the phosphate right down here, okay? Now, of course, the inorganic phosphate is going to be broken down by phosphatase enzyme, Remember, the suffix is an enzyme, right, for phosphate. And so we get this, okay? Now this kind, oh, by the way, the linkage here, the bond that is formed is called a phosphoester bond. And it is a covalent bond. We call it also a covalent bond, right? Now, you're probably sitting there wondering, so what the hell does first of all, Draminors, it's called phosphodiester bond, and you'd be correct, yeah? That's when you have two of them. Yeah, but in DNA, we have two of them, right? Right. Do you know where they are? Listen, a lot of people say and throw around this like it's a slogan or something, phosphodiester bond, phosphodiester bond. Oh, the phosphodiester bond is broken. Now, when you ask them where the phosphodiester bond is, they start, they look at your nose or something, right? Listen, I'm going to tell you exactly what a phosphodiester bond is, and I'm going to explain to you what a covalent bond means, okay? Do you need to know it? Absolutely, you need to know it. You have to know what covalency means. You have to know what the phosphodiester bond is, okay? Okay, and why we call it so. So first of all, let's break down the word. Let's remove bond so we can look at the word in a better manner. So what can you take from the name alone? Phospho, great. So something to do with phosphate, and you'd be correct. And this is the phosphate. What about ester? What's ester? Okay, or you know what? Let's call it a phosphodiester bond. So PDE, phosphodiester bond. Phospho because there's phosphate, di because, well, di means two. E is ester, it's a bond. So, in fact, there are going to be two phosphoester bonds. Now, if you allowed me, I will explain exactly how that happened. Let's remove these here, okay, and start from the beginning, right? Not really beginning, but kind of beginning. Listen, remember when this, here's a nitrogen base, I don't care what kind of nitrogen base it is, but remember when this didn't have a phosphate on it, and it was just a nucleoside, and then we added a phosphate, we said through kinase enzymes, and then we got a nucleotide. Okay, so, and remember how we said that the 5' end here, the 5' end, well, it's a carbon, right? Well, in fact, the chemical formula, the structure of the 5' end is CH2OH. Okay, so it has a hydroxyl group. And the phosphate, here, remember the phosphate has a hydroxyl group also, right? So it was carried by, and it was inserted right here, right? First of all, what does an ester mean? What's the word phosphodiester? Okay, what's an ester? An ester happens when an acid, like phosphate, reacts with an alcohol group, like hydroxyl. The bond that occurs, or the reaction that occurs, is called an ester reaction, or an ester bond, right? Now, remember we said, I told you we were gonna need to differentiate between acid and basic physiologically and biochemical terminology, or the biochemical concept. And here we use it yet again in the ester bond, okay? So an ester bond simply means that an acid, remember we say an acid, the phosphate group is an acid because it donates a proton, so we call it an acid. So an acid like phosphate, right, interacts or reacts with an alcohol group like hydroxyl, okay? It's called ester, or an ester bond, okay? And so you would imagine that when I transferred via the kinase, okay, when I transferred this phosphate group right here, right? So that the hydroxyl group of the phosphate is going to interact with the hydroxyl group of the, well, CH2OH, right? This one here at the 5' end, and this one here are going to react with one another, and again, the hydroxyl group of the phosphate is going to go bye-bye. You get OH, and the hydrogen here is going to go also bye-bye. And so you get H2O. Remember? Because one H came from here, the other came from here, and the oxygen came from here. And this is, again, a condensation reaction. And eventually, let me just do this. What happens is, you know what, here. Easier, right? What happens is, remember, CH2, you know what, let's just remove the carbon, right? Let's just remove the carbon. So, you know, CH2 that was left and an oxygen, right? That is interacting with a phosphate. And this kind of bond is called a phosphoester bond, okay? This is called a phosphoester. Why ester? Of course, phospho, because, you know, the phosphate. Ester, because, again, there was a reaction between the alcohol group and the acid, right, bond. Okay, great. Makes a lot of sense. What about the other bond? The other bond happened when the incoming nucleotide came and basically the condensation reaction happened, and it let go of the two pyrophosphates, and we got another phosphoester bond. Because remember here, it was a hydroxyl group that interacted with an acid, or it was a alcohol that interacted with an acid, and so phosphoester bond. Now, since we have, look, this phosphate group here, it reacted, it made a condensation reaction right here, right? As I made obvious here, right? So it made a condensation reaction here. It made another condensation or a phosphoester bond here. It made another condensation reaction here, a phosphoester bond here also. And so we get two phosphoester bonds. We call it a phosphodiester bond. Okay? But the mic is working. Sorry about that, people. The mic, it died, okay? It doesn't have charge in it or something. Anyway, I hope you can hear me, but just one more point, What is, when I say a covalent bond, right? When I say a covalent bond, what exactly do I mean by covalent, right? It's very, very, very easy. Don't worry about it. Listen, a covalent bond, well, let's look at the word again. Damn, I like words. They give you so much information and you don't have to use a lot of neurons, right? Listen, covalent. Co means together, right? Together. Valent comes from the word valency. Don't worry about it. This is actually more of like chemistry than it is medical biochemistry. But anyway, a covalent bond is a very, oh, by the way, listen, remember when we said that the pyrophosphates are going to be released, the beta and gamma phosphates are going to go, right? And they create a massive amount of energy that makes this bond, the phosphodiester bond, the covalent bond. And that's why DNA is going to be very, very stable, right? Back to covalent bond. Listen. What does valency mean? Or what does a covalent bond mean? A covalent bond simply means that two or more molecules are sharing the same electrons. I know it's not really medical, but listen. Once you get it, you will never unget it. Once you understand this concept, you will never forget it. When you see covalent, you will know exactly what it means. Covalent means strong. It's extremely strong. Okay, so listen, let me give you an example, okay, of something that's covalent. For instance, water. Water has a very strong bond to it, right? Until you boil it and starts to evaporate, then yeah, but strong bond. So, two hydrogen, one oxygen. The oxygen, here's a hydrogen. Listen, here's a hydrogen, and here's an oxygen. Now, the oxygen has 6 electrons. It has 6 electrons, right? The hydrogen, it only has 1 electron. Now, oxygen has 6 electrons, but to become stable, it needs 8 electrons. It wants 8. The hydrogen has 1 electron, this has 1 electron, but they want 2 electrons, right? Now, how do they form water, right? How does the bond occur? Of course, these bonds between the oxygen and hydrogen are called covalent bonds. Now, what happens is, the oxygen here, he goes and he says, listen dudes, how about we share our electrons, right? So I give you, Mr. Hydrogen, I share one of my electrons. Here's 1, 2, 3, 4, 5, 6. I share one of my electrons with you, and you share yours with me. And this hydrogen is like, well, you got yourself a deal, little buddy. And so he shares an electron with the oxygen. And now oxygen is half happy, right? Because he has 1, 2, 3, 4, 5, 6, 7. He has 7 electrons, he wants 8. And he shared this electron, for instance, with the hydrogen. The hydrogen now is completely happy, he has 2 electrons. And then he goes and he talks to other hydrogen. He tells him, listen, hydrogen boy. And that hydrogen's like, what do you need now, Oxy? And oxygen's like, listen, want to share? I'll give you 1 electron, you share with me your electron. And he's like, woohoo. And now oxygen is extremely happy. Why? Because now oxygen, because this one shared 1 electron, and this one shared 1 electron, he has 8 electrons, and he shared his 1 electron with this and 1 electron with this. He didn't lose it. They're sharing. Sharing is caring, people. Listen, 1 electron, 2 electron, 3, 4, 5, 6, 7, 8. Oxygen's happy. What about hydrogen? 1 electron, 2 electron, and this one, 1 electron, 2 electron. So they're sharing. This is called covalent bonding. Okay? And eventually you get something like water. Okay? What about phosphate? What about the phosphate and the oxygen here? Listen, here's a covalent bond, here's another covalent bond. So what's happened? The same story goes over and over again. Oxygen it has 6 electrons, it wants 8, right? So it goes to the phosphate and tells it, listen, Mr. Phosphate, it goes to the phosphate and asks it to borrow electrons. Now, I think, I'm not sure about this, but I think the phosphate has 2 electrons to share, right? So it shares the 2 electrons with this oxygen, shares the 2 electrons with this oxygen. Of course, the oxygen is kind of, it's unfair, it's greedy. It knows that, its electronegative pull is going to be stronger than the phosphate. So we call it a polar covalent bond because basically, it's taking more than it's giving. That's all. Don't worry about polar covalency and non-polar covalency. All I want you to know is that a covalent bond is a very strong bond because of the electron sharing. Okay, so when you hear the term covalent bond, and you're going to hear it a lot, believe me, just know that it has something to do with sharing electrons with one another. If you want to memorize this, memorize it. Be my guest, why not? But you have a lot of things to memorize. Okay? And so yeah. Okay, so let's recap really quickly from the beginning. Don't worry, again, it's not going to take a lot of time. Okay? It's not going to take a lot of time. So, remember at the beginning of this, I think it's even a series by this time, this series of lecture, and we said that we have double-stranded DNA, and then we cut it down, we made it one single-stranded DNA, right? Remember, we had 2 strands, then we had 1 strand of DNA, and then that 1 strand, we took like a piece of it that contained 50 nucleotides. At the time, probably some of you don't even know what a nucleotide is, okay? So this is, let's say it has like 50 nucleotides, and we called it a polynucleotide, and then we took like 15 of them, and now we call, so here it's less than 15 nucleotides, and now it's called an oligonucleotide, and then, we took 1 nucleotide, and we saw that the nucleotide was formed simply of a nitrogen base, here's NB, that's attached to a sugar, which is ribose. Here S for sugar, that's attached to phosphate, and we removed the phosphate, and this of course was called a nucleotide, and once we removed the phosphate, we simply had a nitrogen base that was attached to a sugar, and we call this nucleoside, right? And then, we just parted between the sugar and the nitrogen base, and that's that. Then we talked about the sugar, the characteristics of the sugar, why it's the clinical significance of it. Then we talked about the nitrogen bases and how to tell them apart. Then we talked about the phosphate and the reactions, and of course clinical significance of the adenosine and the guanosine and the cytidine, right? Basically, if you made it this far, you're a goddamn hero. Now, in the next lecture, we're going to be talking about DNA organization. Everything goes downhill. You already did the very hard part. From now on, if you ever get confused, I try to simplify everything as much as possible. If you ever get confused, just go back 10, 15 seconds. I don't think you're going to need more than that, okay? So I'll see you on lecture 2, DNA organization.